this video, I will illustrate the methods I utilized to determine the treatment condition to E. coli cultures via analysis of variation in extracted proteome. These are the contents of the video. The two softwares that have been applied to the analysis are PixX and Panther. PixStudio software is used for LC-MS data analysis and statistical analysis based on experimental design. After the peptide was identified based on MS spectra, the results of the peptide sequence were used to infer the protein composition of the sample. In this study, PixX provided the data sources of protein list with UniProt accession ID, visualized abundance variation output as a heat map, and quality control data of the overall analysis. To further interpret the protein differences in terms of biological processes, molecular functions, and pathways, the data was exported and formatted from PixX, then processed by Panther online platform. The Panther classification system is a comprehensive system that combines genomes, gene function classifications, pathways, and statistical analysis tools to enable biologists to analyze large-scale genome-wide experimental data. As PixStudio and UniProt provided the general biological functions of proteins, Panther overlays the data onto biological pathways. With relatively simple steps, the proteins to be analyzed and their related pathways can be visually displayed through pie charts and tables. For quality control, I selected the data with the least missing value percentage, which are control R1. I opt for the base sample for heat map, and treatment R2, representative for treatments. The curve of FDR-PSM rises steeply as it gets closer to the larger x-coordinate, which indicates that there are more highly reliable identification spectrograms. In this study, the curve of the control group increased in advance around 9,000, but reliable identification spectrograms are still dominated. No major problems were reported in the FDR curve of the treatment group. Figure A is the distribution diagram of score and the number of corresponding spectrograms. Generally, more blue bars with a score of 20 or more indicate a higher proportion of high-quality spectrograms. Figure B is the distribution score of quality score and instrument quality accuracy score, which can be distributed in the range of 2.5 ppm from most blue points, indicating the high and stable detection degree of the mass spectrometer. Both control and treatment groups reported high proportion of high-quality spectrograms, and the brown bars and points indicating error result scores were bounded in low range, so no major problems have been indicated by score distribution. These tables reported the miscleavages proportion of experimental results. 0 miscleavages should be accounted for more than 80%, 1 miscleavages of 15%, and 2 and above should be less than 5%. In this study, both groups' 0 miscleavages proportion is close to 90%, while 2 and above miscleavages is less than 1%, confirmed the reliability of the data. It shows the heat map which selected 70 proteins of the most significant fold changes, excluded 8 of them, the ones in the red box, due to inconsistent expression, and searched their molecular functions manually via UniProt accession code, confirmed their indication with academic literatures, and generated the table on the right-hand side. Then I applied Panther with the exported data from PixX, which visualized the data more straightforwardly and confirmed my researches. Moreover, Panther can also conveniently generate an output of signal pathways relating to the proteins, and simultaneously generate a table include the number of genes classified to the categories, the percent of classified genes to this category over the total number of genes, and the percent of genes classified to the category over the total number of class hits. All those results inspire me that E. coli culture had been experiencing a treatment that caused general downregulation with a variety of proteins. Most of the downregulated proteins, including proteins related to catalytic activity, chemotaxis, flagellar activity, anaerobic respiration, and nucleic acid metabolism, indicated metabolic repression. A few stress-related proteins, which are proteins related to cold shock, acid, and heavy metal metabolism, all showed downregulated status, indicating that the treatment group was less likely to be stimulated by these conditions. Of the 6 proteins that were upregulated, 3 were ribosome subunits, indicating increased protein synthesis. However, more than a third of the enzyme abundance for catalytic activity was reduced. The catalytic activity of bacteria may be related to many factors. Simultaneous upregulation of various proteins could be caused by the lack of carbon required for protein synthesis, combined with the results that sugar intake proteins reduced. ALAC, which was the most significantly upregulated protein, played an essential role in the production of alanine and was directly related to the thickening of cell wall. E. coli cell wall thickening can increase the mechanical strength of the cells, which may be a self-protective mechanism. In the results, the fold change of protein HCHA related to nucleic acid metabolism was 1 to 0.35, which was significantly downregulated. According to Tamla et al. 2013, nucleases in E. coli may be nutrition dependent. The downregulation of this protein verified the previous hypothesis. In conclusion, the E. coli culture in this study underwent shortage of carbon source, which is sugar in common cases. This result is drawn from the general downregulation of metabolism-related proteins, significant reduction of specific proteins indicating nutrition shortage, upregulation of ribosomal subunits, and alanine biosynthesis relating protein. Thank you.